Ja, jetzt kommen wir zum wichtigsten Teil des heutigen Abends.
Now comes the most important part of tonight, namely the speeches, the presentations. All the presentations will be delivered in English because that is the working language of the researchers and because everybody has an international research background. And now I'd like to continue in English too.
With the first presentation, and this is about one of the world's most serious diseases and how it can be detected. Two young scientists have worked on malaria and on a new biosensor which can detect the disease much better than other existing methods. Ladies and gentlemen, please give a very warm welcome to two researchers from the Institute of Biological Information Processing, to Dr. Gabriela Figueroa Miranda, she's postdoc in the group Molecular Bioelectronics, and as well to Dr. Viviana Rincón Montes. She's postdoc in the group Neuroelectronic Interfaces. I've heard you two just completed your doctoral degrees. Congratulations for that.
Thank you. Thank you.
And now you're both continuing your research on a project that will help to fight malaria. Gabriela, of course, that's a huge task for two young scientists, and you already worked so much on this issue, on this research. What keeps you motivated?
What motivates me, so first of all, because I love the research that I'm doing. I love the biosensors research. And second of all, to know that with my research, I can help other people, like detecting diseases. This is what really moves me.
Helping people, I think, is one of the best motivations I can imagine. Well, then I would say please listen very carefully to their presentation entitled Finger Prick with a Big Impact, a Biosensor for Malaria.
Yes, this is the sound of a mosquito. Perhaps for many of you, this is annoying, especially during summer. However, for people living in tropical countries such as in Latin America, Africa, or India, this sound might indicate that you have been infected with malaria. Malaria is caused by the bite of a mosquito that transmits the Plasmodium parasites. Actually, malaria is one of the oldest diseases in the world, with cases dated back to the first millennium before Christ in Greece and China. And despite being known for so long, 229 million cases of malaria were reported in 2019. If we compare this to current numbers of COVID-19, we can clearly see that malaria is still a real threat. Now, if you suspect that you have been infected with COVID, similar, if you suspect that you have been infected with malaria, the first step is to get a rapid diagnostic test. And of course, depending on the result, a specific treatment will be given. However, current rapid diagnostic tests fail to correctly identify malaria. First of all, they often provide false positives. Also, they do not provide a quantification of the parasitemia level. This means how many parasites are there in your blood. And most importantly, these do not discriminate between the different parasites of malaria. And this feature is of huge importance because depending on the malaria parasite, a specific treatment must be given. Now, if we check the statistics, we can see that in malaria-endemic countries, at least 5% of the treatments fail. And in some cases, even 20%. And this is, of course, a serious issue because on one side, we're having a high drug resistance problem, and on the other side, there is an important monetary loss, especially in Africa, where $12 billion domestic product loss are generated every year. Clearly, this can be prevented if we can overcome the weaknesses of this current malaria diagnostic test.
And this is why I started working since 2015 in the development of a high-fidelity malaria biosensor, which was mainly developed during my doctoral research project. However, so far, it has been only tested at lab scale. That means with a big setup and malaria parasite in vitro cultures. Seeing the outstanding performance of this biosensor at the lab scale, I started thinking how to make this sensor really portable and useful for the humanity. And here is what I thought of Viviana, who I met for the first time in a DAD event in 2015, which took place in Bremen. And I remember her since she asked me a lot of questions regarding the presentation that I gave there with the so-called title, Mechanical Minds. And I couldn't have imagined that the destiny will bring us together again. I met her for the second time in Jülich Research Center, where we both worked together at the Bioelectronics IBI3 Institute, under the nice supervision of Professor Offenhäuser. I realized that her background in electronics and microfabrication could complement with mine in biosensing and electrochemistry. So this perfect pairing of knowledge could help us to translate the big and bulky device towards the realization of our groundbreaking device. So currently, we are developing a smaller and portable demonstrator that can be easily transported to remote areas. It is intended to be a portable and user-friendly device detection test, so you can picture it similar to a glucose measuring device for diabetic patients. It is an electrochemical multi-target malaria biosensor. But do not worry, you don't understand all. I assure you that using it is way easier than its complicated name. So it will consist of a small electronic reader and a test strip. And the test strip is fabricated on a flexible polymer substrate, which has several electrodes, and apart from that, it has a low fabrication cost. So, unlike the commercial diagnostic tests that use fragile antibodies, this detection technology uses novel and robust aptamer detection molecules. And aptamers are also better known as synthetic antibodies, since they are artificially selected in the lab, and they are single-strand DNA molecules, so that just like the antibodies bind with a strong affinity to a specific biomarker present in a biological sample, such as the proteins that the malaria parasite produces. So this binding event allows us to detect a signal that we can measure. In this case, in our sensor, we have immobilized 4 different aptamer molecules that can recognize different proteins from the different malaria parasites. Thus, this detection technology allows us to detect and discriminate between the different parasites present in a blood sample. And apart from that, we can also, apart from detecting the infecting parasite, we can also quantify the percentage of the parasitemia, so the amount of parasite present in the blood. We have validated this detection technology in the lab with in vitro parasite cultures. And as you can see here in these 4 graphs, so we tested with the 4 different aptamer molecules, and what we observed, it was a high sensitivity for a low parasite concentration, even as lower as 50 parasites per microliter, with this surpassing the lowest detection limit that the commercial diagnostic test has, and also overcoming the required standards by the World Health Organization. So with this device, the patient can be treated with the right anti-malaria drug and the adequate dosage, eliminating the drug resistance problems and the previously mentioned high costs associated with such failing treatments. Apart from that, we are also implementing in our prototype the system that can allow us to transfer the data to the mobile phone. With this, giving the possibility of epidemiological surveillance and treatment monitoring.
So after these excellent results, we have realized that this base technology of this malaria biosensor can be actually translated for the detection of other diseases. Therefore, we have opened the possibility of having a spin-off so that we can offer this technology as a platform for the detection, either in vitro or in the future, in vivo, of not only infectious but also chronic diseases. As you've heard, we are now developing the first in vitro demonstrator. That means that we are currently looking for funding so that we can finish this demonstrator, and of course, so that we can develop many other applications beyond malaria. So for example, for the detection of COVID-19 and other infectious respiratory diseases with similar symptoms, everything in just one chip. So as you can see, this technology will be essential not only for the detection of novel pathogens, but also as a preparation for future pandemics. It sounds good, right? Well, it's not all plain on research and technology transfer. It is not straightforward to go from the lab to the market. It will take us some time to reach a market-ready product. So starting from getting funding, again, we can bring you then a first in vitro demonstrator so that it can be ready to start a regulatory pathway. And this is important because we have to perform first clinical trials so that we can achieve and obtain the corresponding certifications. So yeah, there is still a long way to go, but it is, of course, an exciting way to go.
So at the same time, we have been also encouraged by different NGOs, such as Foundation for Innovative New Diagnostics, better known as FINE, which work together with the World Health Organization. And they have provided us a positive feedback about this biosensor and even possible help with the preclinical trials. So while the way to go is still challenging, we are currently looking for economical stakeholders to support to grow our project idea, and also for a partner that can complement our team with a business background. Support us, and you will be supporting the transition from novel diagnostics from the lab to the society. Last but not least, we would like to thank all the people that have been fundamental for the realization of this project. Thank you.